So I wanted to start by saying feel free to share the ideas that I present during this talk. If you're using Twitter, I'm PP Gardney, or whatever other social media tools you like to use. And all of these slides are available on SlideShare as well, if you want to grab them. Okay, so I thought I would start by talking about what is RNA. So if you're a mathematician or a computer scientist, you probably think of RNA as a string of 4 characters, A, C, G, and U. And one of the cool things about this string of 4 characters is that inside your cells, it likes to fold up upon itself, and it follows some certain rules to do that. So if you think of the string as different magnets sort of dangling off a chain where you have a north and south pole. In the case of RNA, you also have an east and west pole, and these magnets can sort of attract each other and will bind together. And they like to bind together within themselves, within the same molecule, or they like to bind with other RNA molecules as well. Now, I should mention that a lot of RNA is translated into protein, but there's also an increasing number of RNAs that we know of that are functional within themselves. So functional as RNAs. They don't go through that final translation step. Now, when these things fold back upon themselves, interact with each other, that's not really what happens within the cell. That's just how we project it onto a plane so we can figure out what's going on. In a 3-dimensional cellular environment, these stems, so these sort of stacked pairs of interactions, they like to twist up and interact with each other in fairly specific ways. And the dogma is that this so-called tertiary structure is sort of the functional, the final functional form of these molecules. Okay, so I ran, as Kai said, a database called Pfm for several years. I'm still have a vested interest in this database, so I interact with these people quite a lot still. Many of you will be familiar with the Pfm database. Pfm is kind of the annoying little sister to Pfm. So Pfm was designed to curate protein domains, and the idea was to be able to build probabilistic models of protein domains and use these models to annotate all the protein sequences and discover all the known related domains. Within Pfm, we try to do a very similar thing, but for non-coding RNAs. So what we're doing is we're building alignments with annotated consensus secondary structures of those alignments, and we're using those alignments to build probabilistic models, which we can then search across sequence databases and annotate all the relationships or the homologous families within these alignments. Now the Pfm consortium is headed up by Alex Bateman, who has largely been funded by the Wellcome Trust at the Sanger Institute. Sean Eddy also is deeply involved. He's based at Genelia and is funded by the Howard Hughes. Sam Griffiths-Jones is at Manchester. He receives funding from the BBSRC. And there's little old me, way down in New Zealand, and I'm funded by the Royal Society of New Zealand as well. Okay. So if you're a database person, this is probably how you would view Pfm. So if you're a curator or the curators of Pfm, this is kind of the one little bit of Pfm that we really, really care about. So we spend a lot of time building handcrafted alignments of non-coding RNAs that include annotation of consensus secondary structure. So elements of structure that are preserved across all the sequences within these alignments. And the other thing we do is we write a short description for each of those families that includes links to all the relevant literature and to make sure that we make the most of the RNA community, what we're doing now is we're placing all of those descriptions for each of those families within Wikipedia. So we now have a huge community of people who can go in and keep these entries up to date for us, which is great. Another thing we've done is we've set up a deal with the journal RNA Biology, where experts within a particular domain or experts who are really interested in particular RNA families can write short articles describing their RNA family. And one of the requirements for publishing in this special track for RNA biology is that the authors provide a seed alignment that we can use, and they also need to write a short Wikipedia entry for that, which is great. It means we can slurp all the data produced independently of us and incorporate it more or less untouched into what we're doing. Now, what we do with these seed alignments is we build probabilistic models. We use these probabilistic models to search essentially the entire world of public sequences. So we're using the Emball nucleotide database to do that. On this side of the Atlantic, you probably call it GenBank. And at the time I made the slide, there were around 170 billion nucleotides within this database that we were searching. So once we've found a whole bunch of homologs or evolutionary-related sequences to these families, we can then use an automated process to generate what we call full alignments. So these alignments should contain every member of a family that we could find. And we can use those full alignments to improve the seeds, and other groups use these alignments for genome annotation. So for example, UCSC, Ensemble, and others grab our annotations and present them to the genomics community as well. Okay, so when we're building new families, we're often faced with a slight problem. We heard from Esther this morning, who was talking about quite an exciting technology for discovering new non-coding RNAs. Cole is going to be talking later in the session about another technology that can be used to discover new non-coding RNAs. So the discovery of novel non-coding RNAs is accelerating dramatically, yet often publications don't include the raw sequences within the supplementary materials or secondary structure information in any sort of machine-readable format. So we're often faced with sort of figure one in a paper in a bitmap format that we cannot automatically slurp. Often it is much slower to go and write to the authors and ask for something that we could use. So what I tend to do is start at the beginning of the sequence and type out the sequence and include annotation of the secondary structures at the same time. And once I've finished typing those sequences in, I can then run a homology search, replace all these typed in sequences by hand, and with the best matches, again, within the database, and then build up a reasonably good seed alignment from there. So this is an alignment of an RNA that was discovered by David Hausler's group and was published in Nature a few years ago. I did write a lot of the sequence in by hand. Yes, it was hours of fun. And so this alignment here, you can see some colorings. These colorings correspond to the consensus secondary structure annotation. So sort of this stem here matches with this stem down here. And where it's colored, there's a consensus base pair there. Where it's uncolored, either there's a non-canonical base pair there, or it's in an unpaired state there. Okay, and at the same time that we're doing that, we also construct a Wikipedia entry for each of these families, which hopefully other experts and other interested parties can go in and contribute to as well. All right, so one of the interesting things about curating a large database is you often come into conflict with your user community. So myself as a curator, my number one priority was building lots and lots of new families and try to deal with this accelerated discovery rate that we've been seeing. The other thing I was really worried about was the accuracy of my models, of the alignments and secondary structures that I had. I did not want experts in the field writing hate mail to me because I'd screwed up their publication. I cared quite a bit about the annotation that we're doing. I spent a lot of time working on the code base, the tools that we used to curate and analyze this data. But I spent very, very little time worrying about websites and visualization there. In fact, they were one of my lowest priorities. Whereas if you think about the user community who were interested in the work that we're doing, if you're a bioinformatician, you want to come to a website, find the FTP site, download all the data, and incorporate it into whatever analysis you're doing. You really don't want to spend a lot of time mucking around there. Whereas everyone else wants a nice website, they want nice visualizations of the data within the database, which they can incorporate into their slides for giving seminars. And they were probably a lot less concerned about the sorts of things that I was really, really worried about. So when I started working on RFAM, almost nothing worked. So all the code was broken, the website was broken, a lot of stuff needed sort of revamping and updating. So it was quite a nice opportunity to make some fairly sweeping changes to the database. So I spent a lot of time incorporating quality control code into our family building process. We rewrote the website and we added some additional visualizations of the data that we were presenting. We spent quite a lot of time updating the code base, making it a lot faster, a lot more robust. And at that time, we also exported all of our annotations to Wikipedia and tried to drum up more support from the RNA community by setting up this deal with the journal RNA Biology. So when I was thinking about what sort of visualizations I wanted to include, my number one priority was scale. So if you have the nicest visualization tool in the universe, but it only runs on 2 sequences, an alignment containing 2 sequences, then I really couldn't use it. Because the alignments in our database can range from just 2 sequences all the way up to 2 million sequences. The other thing we're dealing with is some of our alignments are very short, so only sort of 30 nucleotides long, but others are extremely long. So for example, the long subunit of the ribosomal RNA is around 30,000 nucleotides long. Some of our consensus secondary structures contain no base pairs, others contain up to 1,000 base pairs as well. So your tool really needs to be able to deal with all of these issues before I could consider using it. It also needed to be automated as well. So we had thousands of families in the database. I was not going to interfere manually at any stage during the construction of any visualization. The other thing I really, really wanted out of the visualizations that we were generating is something that was informative. So if your tool is really, really pretty and really nice, but provided absolutely no novel biological insight, then I really wasn't interested either. But if your tool satisfied these 2 criteria, then I really wanted to be inclusive. We really have a really nice bioinformatic and visualization community, and I didn't want to piss anyone off by leaving them out. All right, so I'm going to show a few examples of the sorts of things we want to be able to visualize at RFAM, with the caveat that none of the images I'm going to show should be considered final solutions. Just about everything I'm going to show can definitely be improved upon. I'm going to show an example of a secondary structure for starters. So this is a secondary structure for a CD box SNO-RNA. So if you know anything about CD box SNO-RNAs, you can look at this visualization and you should be able to see almost immediately that it is one of these. So this is a cartoon representation of one of these SNOs. They often have a short stem, which corresponds to here. They have two sequence motifs, a C and a D box. Now the colors here illustrate evolutionary conservation. And what's really cool about this visualization is you can see immediately that these two highly conserved regions correspond exactly to the C and D box motifs. So these motifs are involved in RNA-protein interactions there. So you can see that evolution is also preserving these. The other thing that's important to know about CD box SNO-RNAs is that they go off and bind to other RNAs, so they interact with those as well. And you can see down here we have another conserved region, and that corresponds exactly to the region that is binding to another RNA too. So there are sort of many nice things about this visualization scheme. There are some not so nice things as well. The color scheme is pretty garish, and the layout can often be a little bit nasty as well. So improvements can be made, but it doesn't seem to be too bad. So the other thing one of the other sorts of data we want to be able to present is taxonomic distributions. So for each family, there are between sort of 1 and thousands or millions of species where these sequences are found. Now this is a non-coding RNA called RBB, which Gigi Storz, I believe, was involved in discovering. And Gigi can probably tell you what species she found RBB in. It was found in E. coli. Now, our old tree-based method for displaying taxonomic distributions had several flaws. So one flaw is that you had to scroll quite a long way down a list before you could really get an idea of what the real taxonomic distribution was. And in this case, it went horribly wrong because what can happen when you're dealing with public sequence archives is people can mislabel their sequences. So in this case, an E. coli sequence has been labeled as a eukaryotic sequence from this nematode species, which I'm probably not going to be able to pronounce. And basically to our user community, the first thing we were presenting to them was contaminant, or false data, which was unpleasant. And then after our user had spent a bit of time scrolling, they eventually found the correct species distribution there. So we've since replaced that tree view. There were some other bad things. It was really, really slow to build as well, so it hit the website pretty hard. We've replaced these with sunbursts, which are related to pie charts. I realize in this community, pie charts is probably a dirty word, or dirty 2 words. But in this case, it's certainly a hell of a lot better than that, I would argue. Too far. Now, what the sunbursts are showing, so at the middle of the diagram is the root of all life. This sort of first arc is the domains of life, so archaea, bacteria, eukaryotes, and the length of each arc. So this word here, which you can't read, actually I can't read it either, is bacteria. And you can see that the vast majority of the sequences are in fact bacterial. And as you work further and further out towards the outermost set of arcs, these correspond to the species there. So this big arc here, which runs from all the way up there, all the way around to here, corresponds to the E. coli sequences. So you can see immediately that this is predominantly an E. coli family. And those awful mislabeled contaminant sequences that I was complaining about before, they're just this very small wedge here, which hopefully most people can ignore. Okay? So the other thing we spend a large amount of time looking at are alignments. Basically, where you're sort of stacking sequences on top of each other, and you're looking for conserved columns within your alignment. So basically, what signals is evolution preserving for us there? And we spend quite a bit of time worrying about the secondary structure annotation here, which we've illustrated with open parentheses and closed parentheses to annotate matching base pairs. Now, one of the things that really worries me about the future is when we have sequenced every organism on the planet, my alignments are going to be ridiculously large, right? They're not just going to be tens of sequences deep, they're going to be millions of sequences deep. So what on earth are we going to do then? I think we're going to have to start to think about this probably sooner rather than later. I'm looking at you, Jim. We need to be thinking about ways to partition the data cleverly, and probably data structures that are going to help us do that sensibly. The other thing I'm extremely interested in is genomic context. So for any non-coding RNA, where it lives on a genome sequence can tell you a lot about the function. So if this non-coding RNA happens to be in the middle of an operon, so here I'm showing gene annotations for Salmonella typhimurium. This is an operon, so a group of functionally related proteins live in here. If we happen to have a non-coding RNA in the middle of that, and that context is preserved, then that can probably tell us a lot about the function of the non-coding RNA, which is a major hurdle to get over when dealing with RNAs, is developing hypotheses as to what the function is. Now, this is a figure that we published a few weeks ago in Nucleic Acids Research. And so this sort of order of the genes along the genome is preserved between these two bacteria. And with these sort of gray boxes, we're trying to illustrate homology, so evolutionary relationships. But you could also think about replacing this with sort of gene ontology relationships or metabolic pathways or anything like that. Now, this sort of view works great for just 2 species. It's not too bad. But what I'd really love to be able to do is to do the same thing, but for hundreds or thousands of species. So, is this gene order preserved across all bacteria or across a specific clade of bacteria? And if this gene order is preserved, then what can that information tell us about the function of non-coding RNAs? So if anyone has any ideas how to do that, I'd love to hear from you. All right, so I think we still have a number of open problems to address. How do we display evolution and RNA structure? Is there any way our alignment visualization and editing tools are going to scale? Can we include information about expression levels and conservation levels? And in particular, I think it'd be very cool to be able to look at genomic context within an evolutionary context. Many thanks to all the people I've worked with over the years within the RFAM consortium. We also have a fantastic community of people who are developing visualization tools for looking at RNA, and we have some great funding agencies. And this is the view out of my office window, if you ever want to come and visit.